Thank you for Rene and Martin for joining us today and talking to us about comprehensive comparative analysis of primary photophores and lanternfishes. I am not a fish person, so I'm excited to learn about a new topic for me.
Perfect, yeah.
Welcome you and thank you.
Thank you so much. I feel like I've met most of the people in this room, which is pretty great considering I just started in August. But if you don't know me, my name is Rene Martin, and I'm one of the new faculty members that recently got hired here in the School of Natural Resources. And I'm actually going to be talking to you about deep sea fish. That is what my research background is on, which is kind of weird being, here at the School of Natural Resources. And so most of the work I'm going to talk to you about today is actually associated with the work that I did in my postdoctoral position at the American Museum of Natural History. But before we do that, I have to talk to you about the deep sea because we are inherently biologists from the Midwest in a landlocked state. And so you might not have as familiarity as I do with the different factors of the deep sea and what goes on there. So we're going to start with a basic introduction to this area, and then we're going to move into my research after that. So the deep sea is made up, historically actually has been divided up by scientists into different zones. And I'm going to be focusing on the pelagic areas, which are those open ocean areas, and not the areas associated with sea bottoms or continental shelves and things like that. So we break them up into these different zones, the first and the topmost being the epipelagic zone. So the epipelagic zone is from the surface down to 200 meters, and it is not considered a part of the deep sea. So this is the photic sunlit zone. This is where all of the zooplankton and phytoplankton are and where all that photosynthesis occurs. But below that is when you get into the deep sea. So this mesopelagic zone is that next 800 meters. So it goes from 200 to 1,000. And this is what we consider the beginning of the deep sea area. And it's an area where you start to lose sunlight, you start to gain pressure, it starts to get cold. And then continuing that deep sea area, you move into the bathypelagic zone and the abyssopelagic zones. And so these are all the deep sea. And then once you finish that mesopelagic zone and below, you have no light at all. Nothing from the surface of the ocean can continue past the mesopelagic zone, even on the brightest day. And to kind of give you an idea of what these depths actually mean, if you took the Eiffel Tower, for instance, and you put it from the surface of the ocean down, it would just reach into that mesopelagic zone. If you took the Burj Khalifa, which is the largest man-made building on the planet, it would just almost reach the bottom of the mesopelagic zone. And if you took the entire length of Central Park, it would reach down through the mesopelagic, almost all the way through the bathypelagic. And prior to actually moving and living near New York and being at the AMNH, I actually didn't really fathom how long that was. And so it was kind of nice because I could walk the whole length and be like, that is the deep sea right there. And so also to give you an idea, the average depth of the ocean floor is usually between 4,000 to 6,000 meters. So this excludes trenches. Trenches can go further. But the average depth of the deep sea is right around this. There's not a ton of area that are abyssopelagic. And this deep sea area, compared to the terrestrial systems that you guys are really used to, has very static abiotic factors. Because when you're here in Nebraska, we don't have big bodies of water. We don't have things that can control temperature. You've got huge fluctuations in temperature. You've got wind. You've got all sorts of stuff. And that just doesn't occur in the deep sea. And so to give you an idea of what's going on, you have temperatures and abiotic factor that is dropping dramatically in that first 1,000 meters. But once you pass that, you have a very static cold temperature for basically the rest of the deep sea. If you're looking at pressure, when we're here, we actually don't think of pressure as something that is like affecting us on a daily basis. But in the deep sea, you have hydrostatic pressure that increases very linearly with depth. So as you are a creature and you're living deeper and deeper and deeper, that pressure is linearly getting more and stronger and stronger and stronger, and you have more pressure on your system. If you are thinking of oxygen, like unless you're studying something in the mountains that might have low oxygen, we don't really think about that as much. But in the deep sea, you have high oxygenation right at the surface of the ocean where you have wind and wave action and all of that water is getting more oxygen, but you have this steep cutoff of oxygen right here in the mesopelagic zone, which is kind of that mid-twilight area. And these are called oxygen minimum zones, and things have to move through that area quite frequently. And they have to figure out how to get through an area where they have little to no oxygen. But what's interesting is you have this increase in oxygenation in the deeper parts of the ocean. Cold water can hold oxygen better, and what ends up happening is you have polar ocean seas that have oxygenated water that drops and moves along the ocean floor and brings this cold oxygenated water to other ocean basins. And then another abiotic factor that we consider when we think about the deep sea habitat is light. So again, you have light at the surface. It penetrates all the way down to about 1,000 meters, and then beyond 1,000 meters, there's no light that comes from the surface of the ocean. So I've just talked to you about all the abiotic factors, or at least some of the major important abiotic factors of the deep sea, but there's one truly major biotic factor that structures this open ocean interconnected area, and that is something called the deep scattering layer. So the deep scattering layer was first discovered by sonar operators in World War II, where they'd be going along, they'd be using their sonar system, and the system would bounce back and say that the ocean floor was about 400 meters in the area that they were looking at, which is totally fine. There's nothing wrong with a 400-meter ocean floor. But this would be in areas where they knew that the sea floor was 800 meters. So why were they getting these readings? And what was interesting is that at night, they would be still using their sonars, and that ocean floor would actually move closer to the surface of the ocean. And so like, well, what's going on? What is happening? Why are we getting these false sea floor readings? And what they found out is that there's trillions and trillions and trillions of organisms that exist in these large swaths that cover hundreds of kilometers and can be meters and meters thick that are all a part of this vertical migration. And so they consist of things like fishes, and these fishes have these swim bladders. And so swim bladders help them keep buoyancy in the water column. And what was happening is these sonar readings are bouncing off of the gas inclusions in swim bladders and then bouncing back. And there's so many organisms in these swaths of migrating like packs that you get a false C4 bottom. And it's not just fish. There's all sorts of things that live in these migrating layers, including things like Photophores and squids and crustaceans. But you have this really integral biotic system that structures an open ocean area that is connected globally. And it is one of the major factors of and major habitats of the open ocean deep sea area. And so why are they doing this? Why are they moving up to the surface at night? Well, it's thought to be due to predation. So up in those surface areas you saw, you've got lots of sunlight, you have lots of phytoplankton, and what eats phytoplankton? Zooplankton. And so you have the habitat of the surface of the ocean creating a bunch of food sources. And so it's believed that these deep sea fish and other things are swimming up to the surface of the ocean at night to feed on all of this zooplankton. But why do they not stay there, right? Why wouldn't you just stay at the surface of the ocean, especially if there's less food sources in the deep sea? Well, there's a lot of predators that live in the surface of the ocean. And so we think that they migrate down further into the deep sea during the day because they can escape from the visual predators of the surface. And so they do this daily migration every day. And it's thought to be actually the largest migration on the planet on a daily basis. And so you would think, hey, we've got all these deep sea fish and they are swimming up to the surface of the ocean to eat. Why do we have so many of them? Why are they so diverse? Is it because they, ancestors lived near the surface or lived near the shore and they were pushed into the deep sea and had to live down there because they were just pushed out? There was less food sources, and so they have to scrabble up and eat every day and swim back down. Like, how did all of these fish that live down here evolve? How did they come to be? And what is interesting is there's actually a lot of evidence that shows that these fish weren't like forced into the deep sea. You have a lot of extremely diverse lineages of deep sea fishes. including things like the dragonfishes, which there's hundreds of species. The lizard fishes have moved into the deep sea multiple times, hundreds of species there. Lanternfishes, which I'll be talking about more, hundreds of species again. And you can see on this phylogeny that the ancestors of all these groups very much likely evolved in the deep sea. And so they weren't being like at the surface and have a bunch of independent, fish moving down there and living there and evolving there. You've got ancestors living and evolving in the deep sea. And so how do we get such diversity down there? How is it that we have all of these deep sea fish that live in an arguably very difficult to live in, dark, low biomass food item area? And there's kind of one major hypothesis, one major reason, and that is bioluminescence. And so bioluminescence, as we think of here in the terrestrial land, you've got fireflies that can glow. It is a very similar system, at least in the chemical compounds. It's where you have this luciferin and luciferase and oxygen, and those combined create light. And this light is useful to some of the organisms that live in the deep sea area. But how does light and how does bioluminescence itself help diversification, help these different organisms speciate? Well, light is used for a lot of different things. And I have a lot of pictures of a lot of dead fish, so we're going to show you some cool video bits of bioluminescent fishes too. So this is a bioluminescent dragonfish. It was taken actually, I think, by the sponsor that I worked with at the AMNH. And bioluminescence is used in a lot of different ways. It can be used for things like defense, offense, communication, all of it. And depending on how it is used, can actually affect whether or not something may be speciating due to its use. And I'll get into that a little bit more. And so one major thing that some of these deep sea fishes used and has occurred and evolved countless times in fish that you see that live in the deep sea is something called counterillumination, which is this guy right here. And so counterillumination is when you have a fish that has the light organs that are on the ventral surface of its body. And those light organs can match the intensity and the color of downwelling light that is filtered through the ocean. And this downwelling light tends to be this blue color. It's like a blue teal because those are the wavelengths that can penetrate the furthest. Reds get scattered out, yellows and oranges get scattered out, and even greens get scattered out until all you have left is this blue color. That's why when you have things that are filmed underwater, they tend to be blue. It's because all the other colors get scattered out really fast. And so if you have light organs on the surface of your body that you can match the color of that light that's coming down, it obscures your silhouette from predators that might be lurking below you in the water column looking to eat you. And so a lot of the organisms that can do this do counterillumination, that are using bioluminescence as defense, which is great. I think that's fantastic. It is a really, really good way to promote survival in this deep sea area. But you can also use bioluminescence for other types of defense, like the prize and smokescreen. And so this is a cardinal fish that's about to eat a tiny little ostracod. And so he eats it, and then this ostracod makes a bioluminescent bright light, and the ostracod, or not the ostracod, but the fish spits them out really quick, and then the ostracod can either swim away or the prey item can escape because the predator has been blinded slightly. And so this is one way that they also use it as a defense mechanism. But you can also use it as offense. So we know many of these offensive measures, and so I'll just talk about a couple. So these are flashlight fish. And flashlight fish have bioluminescent light organs right underneath their eyes. And so their eyes are actually above that light organ. And a lot of deep sea fish that use bioluminescence have light organs under their eyes. And it is thought to be used as a way to either search for food or to flash a bright light at a predator or at a prey item so you can distract it, blind it, and then eat it. And I mean, as many people know, we've got anglerfishes and different types of lures. So anglerfish aren't the only fish that have lures. Some fish have lures on their chins, some fish have lures on their heads, but all of them are believed to be used for a similar purpose, which is luring in a prey item. There we go. Luring in a prey item. So why would a lure work this way though? Why does a light attract these fish? Well, a lot of the zooplankton that I talked about earlier are actually bioluminescent. So they can create bioluminescence themselves. And so if you have an anglerfish that's swimming along and its lure matches that of a prey item, which would be a tiny little zooplankton, he might be luring in another fish, which he can then eat himself. And so it's not until we get to sexual selection and communication in which we think that bioluminescence may be helping these groups of fishes diversify. All of these other behaviors are useful for survival, useful for all sorts of things, but it's not until we see communication, sexual selection, and species recognition that we think that these groups are aided in their diversification because of bioluminescence. And that's because you can potentially identify each other with lights and with communication. And so here is just one example. These are ostracods, and they themselves are ahead of all of these paths. And so when they're doing their courtship displays, they're moving along and they're making a bright light as they move. And that light, akin to what fireflies do, is very species specific. And so males and females of the same species can actually find each other during these courtship displays because those lights are an indicator of, hey, that's the species, that's the male that I want. I don't want that male, he's flashing the wrong pattern. And so these are really, really important for our understanding of how some of the organisms that use bioluminescence in communication diversify in the deep sea. And so we've seen this multiple times across many different groups, including fish, including, invertebrates, all the invertebrates, where you have these lineages that have bioluminescent courtship displays or bioluminescent sexual dimorphism that have higher species richness, higher diversification compared to their sister lineages that are either non-luminous or don't have those sexual displays. And this happens in fish too. So within fishes, bioluminescence is actually believed to have evolved about 27 different times along the tree of life. And just to give a little bit of a caveat, intrinsic versus symbiotic. Within all of these groups, fishes included, invertebrates included, bioluminescence can occur in two different ways. One way is to have a symbiotic relationship with the bioluminescent bacteria that exist in the open ocean. So they exist anywhere. You've heard of like the bioluminescent tides, you can splash around on the water. Those are bioluminescent bacteria. And what happens is a lot of these fish that have evolved in these areas have evolved the ability to kind of house those bioluminescent bacteria and create a really good system for cultivating them in their bodies. And then they use that as kind of a light organ. So those flashlight fish, the anglerfish, all of those are the bioluminescent bacteria. But some, including lanternfishes, which we'll get to, have intrinsic bioluminescence, where their light organs are actually innervated by nerves. They have the cells that can do this, the cells that can light up. It is believed that they get some of that chemical, so the luciferin and luciferase compounds, from their food. So they might have to go and eat zooplankton to get at least one of the chemicals to produce bioluminescence, but they can still do it intrinsically. And so within these groups that have sexual dimorphism in their bioluminescence, whether it's in their light organs or in their flashing patterns and displays, within deep sea fish, we have higher species speciation and diversification within these groups. And so it kind of gives us a good idea, because we have so much evidence that bioluminescence plays a really important role in a lot of these groups in diversifying in the deep sea. And so that gets me to lanternfish, now that you have a little background on deep sea. So lanternfishes are within the order Myctophidae, and they are the group that I've been studying for the last 7 or 8 years now. And within the Myctophidae, it's actually a really cool group to study because there's 2 families. The first family being Neoscopelidae. Neoscopelidae is a very species depauperate group. They're called the black chins. There's only 6 species. And what's interesting is one of the genera, so there's 3 genera, one of the genera has bioluminescence, but that bioluminescence is only that counter-illumination of ventral photophores. They don't have anything else other than that. And that's compared to their sister group, which are the lanternfishes. And so here are the lanternfishes, over 252 species, and numerous accounts of sexual dimorphism in their light organs. And so we see this group that has 2 majorly different amounts of speciosity, diversification, whatever you want to call it. And so they're really cool to use to compare the differences between the 2 groups. So again, just to give you a highlight, we've got one group called the black chins, Nebraska-Lincoln, only about 6 species. They are kind of pelagic, but mostly demersal, so they're mostly associated with the ocean bottom, but they often go into the water column, have bioluminescence, but only on the ventral portion of their body. And then you have lanternfishes, 252 species worldwide, and more pelagic. So you don't really see these fish associated with the bottom unless they have migrated and kind of hit the bottom and they're like, oh, I hit a seamount, oh no, and then they'll like swim back up during the next migration. And what's interesting about lanternfish themselves is they are thought to be one of the most abundant vertebrate groups on the planet, like truly. Studies that have looked at their abundance across the world's oceans, they trillions of tons of these fish across every single world ocean, which I find fascinating because I didn't know what a lanternfish was prior to studying them. They are, again, also bioluminescent, but that bioluminescence occurs in both counterillumination and in different patches and different organs on different parts of their bodies. So, like their name suggests, here's the organs right on the ventral surface thought to be used in that counterillumination. Even in this picture, you can see that they're kind of blue, matching that downwelling light when they're turned on. But like I mentioned, they also have additional light organs. So oftentimes they'll have these accessory light organs that are located under the eye and also on the tail. And then they also have primary light organs that have moved up to the side of their bodies. And those primary light organs are actually very species specific. So if you look at the ones on the ventral portion of their body, they're not really species specific, but if you start looking at the ones on the side of their body, we actually use those very frequently to determine what species that is. And since you're going to see a lot of dead fish, again, here is a freshly caught lanternfish. They usually have very, very dark black bodies and very bright, shiny, bluish scales or silvery scales. But when you catch them in trawls, they're really delicate. So their scales will fall off, their skin will disappear. And unless you've netted them at the surface as they moved and did their migration, it's very likely that the specimen that you get is going to be just torn apart. So there's an example of at least one pretty one. Enough of that. So similar to all of the studies I've mentioned before about how these organisms that are bioluminescent and use bioluminescent for communication in the deep sea or sexual selection and species recognition, lanternfishes also have been found that they have higher rates of diversification and speciation compared to the age of their clade. And so this is believed to be because of the fact that they have and use bioluminescence for communication. Now, we are only inferring that they use it for communication because we can't know. We can't, like, it's been very difficult to go down into the ocean and film these things because we just don't really have the cameras and the facilities and the abilities to do it. And once we do, it's very expensive. So that technology is increasing, and hopefully, you know, in the next 20 to 30 years, we'll be able to verify a lot of the things that we are inferring because of this. But for now, we believe that they are using some of their light organs in order to recognize each other and to potentially isolate themselves as different species. And the reason this is difficult and the reason why it's a weird and interesting topic to look at, especially in deep sea fish, is when we think about speciation in terrestrial systems, we think about allopatric speciation, where you have two populations that have diverged and they're just isolated from each other. And potentially, if they come back together in the future, they've been so isolated and have changed so much that they just can't reproduce together anymore, which isn't the case in the deep sea. You have this open ocean barrier, which has no major barriers to gene flow. And so how are you getting 252 species? So again, we think it's bioluminescent. And not only that, but lanternfishes themselves, their eyes are extremely attuned to see the specific wavelengths of bioluminescent light. We are able to see a lot of different colors. Lanternfish are almost explicitly able to depict and pick out different wavelengths within those blues and greens. And not only that, but some of the individual species, males and females, have these, they're part of their eye that actually filters out this blue color that may, what's the best way to describe it? It may help them depict differences, slight differences between species in bioluminescent color, and males and females have sexual dimorphic differences between these these eye capabilities. And so why would you have sexual dimorphism in your ability to see a certain wavelength of light unless it was potentially useful in discriminating between different colors of bioluminescence? And so there's just a lot of work that's been done just to kind of look at and to hypothesize as to what's going on here. Which leads me to my work. So I've studied lanternfishes for a while, and going back to 2018, one of the first things that I did for my master's was to create a phylogenetic tree of lanternfishes. And phylogenetic trees are really good for understanding a lot of different things because you can use the relationships that you infer as a backbone to look at how a trait has evolved across the group. And so in this particular case, I used 79 different species and a bunch of different types of data to create this phylogeny of the relationships within lanternfishes. And so what I can use this for and what I can do is I can then look at and I can analyze and study different traits of each of these different species. And then I can plot those on the tree and I can look to see how it's evolved. I can see whether or not the fact that they're just related to each other or the reason they have a certain trait, or if it's actually habitat that's selecting for certain things. So phylogenetic trees are really important to my work and important to a lot of work. And so this was the first step in a lot of what I did. Maybe. I use those up. And so to move into the work that I did at the American Museum of Natural History, it was to better understand how light organs work and have evolved within lanternfishes. Because this is clearly a very important trait within this group. And so what I did and what I want to kind of point out to you is we're just going to go a little bit more in depth into the two different types of light organs. And so lanternfishes have these things called accessory light organs and then photophores, but I'll dive into photophores in a little bit here. These accessory light organs are found around the eye and also around the tail. Some species have patches that are located on different parts of their bodies. And these light organs are actually considered simplistic. So they are these stacked cells, and all of these cells within this distinct system are multinucleated and they are light producing cells. So it's basically just a big stack of light producing cells, which we consider to be somewhat simplified light organ. But what's interesting is that although it's a simple light organ, the shape of these light organs is very variable. So this is the light organ that you see sexual dimorphism in, where a male will have a really, really big one, but a female won't have one. And so this is the one that we believe is really important for communication. And the reason that this particular, especially the tail light organ, is important is recent work that have looked at all sorts of things associated with the body shape of fishes found that fishes that live in the deep sea specifically have a relaxed constraint on their caudal peduncle. So the caudal peduncle is the tail right here. And the reason the study says so, or what their hypothesis is that because the fish that live in the deep sea don't have to worry about swimming through strong currents, they don't have to worry about swimming around coral reefs or things like that, that they actually, their body shapes are much more able to just kind of evolve with all sorts of other random things. They're not as highly selected for the conditions and the habitats of things that are near shore or more coral reef based. And so you have a relaxed constraint, which is kind of cool because I have this group of fish where you have a lot of individuals that have a caudal tail light organ. And so is it possible that this caudal tail light organ may be affecting the morphology of that caudal peduncle and the body shape of lanternfishes because they now have this relaxed constraint on their body morphology because they don't have to worry about all the things that the fishes of the surface and the nearshore do. And what's really great is that within lanternfishes themselves, we have all of these different species, and Lamponictinae and Myctophidae all have these caudal light organs. But I have a fun group nestled within lanternfishes, which is Diaphinae, that do not have caudal light organs. And so I can actually test to see if there's differences in the body shape and the variation of bodies within this non-caudal light organ bearing group compared to the rest of lanternfishes, which is great because this group is also the most speciose group. It's got about 80 species in it. So in order to do that, I did something called geometric morphometrics, where I analyzed like 1,000 different species. I took pictures of them and I placed landmarks across their bodies. So these white ones are homologous landmarks. They're on a very specific point. So, insertion of the dorsal fin, insertion of the anal fin, et cetera. And then the semi-landmarks are curvature landmarks that are associated with curvature of the body. So they can kind of shift a little bit depending on how the body increases in depth or decreases in depth, et cetera. And then what I also did was I wanted to see whether or not this caudal area evolved differently than the rest of the body. And so you can break these up into different modules as well to make comparisons between how shape is changing in the bodies of these fish. And so I did that with, I can't remember, over 150 species within lanternfish, which is pretty good coverage. And I found that, so this is a principal component analysis for those of you that aren't 100% sure what's going on here. The center of this plot is the average lanternfish body shape. And around that, you've got principal component 1 and principal component 2. And these little graphs here show kind of what the extremes are of the edges of these graphs. And so you can see down here, you've got shortened, more truncated bodies. Over here, you've got slightly elongated bodies. And then up here, you can see you've got differences in the caudal peduncle and the location of that anal fin insertion versus down here. So it just kind of gives you an idea. And so you can see that this subfamily, Myctophidae, which has really, really robust, cool, different shapes, different sizes of not only its body, but its tail light organ, is also the most disparate in its body shapes in lanternfishes, as opposed to Diaphonae, which has arguably more species, much more constrained in its body shape. And so this is just with the phylogeny plotted onto that graph as well. And so you can kind of take a look too that within each subfamily, you have these fish that actually their body shapes move in different directions in morphospace. And again, like I mentioned, you have Diaphonae, which have all converged on this end of morphospace. And not only that, but I added that subfamily, or not subfamily, but the other family, the black chins, also don't have light organs, are also associated with this corner of morphospace. And so it's thought that maybe they have converged in this area. And if not converged, it could be that all of these other species that have those caudal light organs and are potentially being selected on those caudal light organs because they're being used in communication, that relaxed body has allowed these body shapes to be much more variable in the deep sea compared to the ones that don't have selection on the caudal light organ, where they're maybe more constrained or just have had no reason to vary in their body shape. So that's all the work I did, or some of the work I did prior to the American Museum of Natural History. So we talk about those accessory light organs, but we're going to move into photophores themselves. So photophores are the light organs that are associated on that ventral surface of the body, but in lanternfish, a lot of them have also moved up onto the lateral surface of the body as well. And like I mentioned previously, those are species specific. So oftentimes, if you're looking at a key to the fishes of lanternfishes, you are using the number and the placement of these lateral photophores to identify them, which we think potentially is useful for species discrimination as well, but that's up in the air. We're not 100% sure. But what's interesting about these photophores is that they actually have been found to emit slightly different colors. So you've got some in the blues, you've got some in the whites, you've got some in the purples, some in the yellows, slight differences in color. And so how is that happening? How are these fish doing that? Especially if the photophore itself is kind of an ancestral character to the entire group. What is making these changes? And that is what I kind of set out to at least start investigating when I went to the American Museum of Natural History. What is it about these photophores across these different species that enable this different color? And so I looked to see what other species have had their photophore anatomy, microstructure investigated before, and found that there was really only about 13% of lanternfishes that have had their photophores investigated. And not only that, but most of the work that was done in the early 1900s, late 1800s, early 1900s, which the people that were doing the research back then did a fantastic job, but also you can only work within the realm of like what your microscopes can allow you to see things like that. And so a lot of this morphology was described really, really early on and kind of left for a really long time. And it wasn't until more recently that we finally get actual images of the histology and anatomy of some of these light organs in slightly more recent papers. But even since 1973 up until the present, there's really only been 4 or 5 papers that have looked at light organ structure. And so that begs the question of like, okay, I think this can be investigated more. There's 252 species of these, and it actually hasn't been described in many of the genera themselves across the group. And for those of you that have looked at 2 different species that are in even different genera in fish here in the Midwest, like there can be pretty significant differences. And so this hasn't been investigated across lanternfish. So to give you a little idea of some of the structure, photophores have, so this is the outside of the body and it's got scales. Each photophore has a scale lens, and that lens is a modified scale that's been thickened and acts to direct light. You have photocytes, so the things that produce the light. And then closer inner to the body, you've got something called a scale cup. You have a pigmented layer, and then between the scale cup and the pigmented layer, you have a reflector. And so that is like a reflection disc that any light that goes towards the body is bounced and angled out into the ocean. And you can see that the scales that are overlaying these may actually affect the color. So the 2 starred scales here are the ones that have a scale overlaying it, and then these 2 have been lost. And so you're seeing potential differences in color, and we haven't really explored or investigated this again across the group. And so that is what I did at the American Museum of Natural History, was to try to fill in the gaps of knowledge and to investigate the anatomy of the photophores of lanternfishes, which says right there. I also wanted to look at the scale lens. So this thickened scale lens, there was hardly any studies that had looked at it. And it's a really important component because it is the last thing that the light comes through as it enters out into the ocean. So is there any variation in that scale lens? If there is, that can actually affect how light is attenuated out into the ocean. And then use that data, compare it to some of the historical documents on species, and just kind of do a broad swath of investigation with the future goal of looking at this within a phylogenetic context. And so just for this work, I looked at 24 different species from a couple different institutions, including the American Museum of Natural History, the Natural History of Los Angeles County, and then MCZ at Harvard. And I removed the light organs from this VO series right here. And the reason I did that is because there's a pelvic fin right here. And this pelvic fin often protects the scales from being lost when this fish is collected. And so that VO series, generally, if it was going to have a scale still attached, it was going to be on that VO series. And then I did a bunch of different staining methods using histological methods. So if anyone's interested in learning that, we're slowly gathering all of the equipment in my lab here. And so histological methods to look at the anatomy and the structure of these light organs. And so again, I looked at some of the other genera that had been investigated before from these previous works to both compare and to add to the data set as well. So what did I find? A lot of variation, but also a lot of similarities, which is to be expected. They are all the same thing, which is a photophore. And so you finding variation is great, but finding similarity is expected as well. And so to orient you a bit to the histological slides, you have this really thin blue area. That is the reflector, so the reflecting disc, which the light comes through this red stained thing, which actually in life would be a clear photophore cup. You have this block right here of photogenic cells. And so there are these stacked lamellae, like I had mentioned in those more simplified light organs, but here you can see them in the center. And this is just connective tissue that holds it all together. And then usually have this pigmented mantle, which this pigment prevents light from entering the body. It's just kind of a barrier. And I saw a lot of variation. And so you can see the variation either occurred in the amount of photogenic tissue compared to the size of the photophore. Some of this variation was in the thickness of the photophore cup. And some of this variation occurred in multiple layers of reflective layers. And so I'll get into the pattern on this a little bit more. While looking at that modified scale lens, just again, to give you an idea, you have calcified scale lens and collagenous scale lens. And in my study, I found that most of the scale lens, which is the red part here, was calcified, and that you have a thin layer of kind of a collagenous scale lens, whereas in some of the previous studies, they had had it flipped, and so further investigation on that is still needed. But I found that most of these were pretty calcified. And so here's an example of some of the variation that I saw. And these are all stained exactly the same way. And you can see that in some species, you have very calcified scale, whereas in others, you have an area of collagenous material prior to the calcified matrix. And again, light is coming in this direction. And in some, you had kind of a mixture of both. So we're seeing also some variation in the shape as well, where you've got very extreme tapering on the ends, and then you have a more teardrop shape, modified scale lens, and then others you have much more or much less drastic tapering. And so just looking at some of the patterns, what we noticed and what I noticed is that the scale lens and the scale lens morphology, or not scale lens, but the pigment layers and the reflective layers in this whole entire group here were really, really hard to see. And so this is like gymnoscopelinate, lamponectinate, and diaphanate. It was really difficult to discern some of those layers. And some of it could be just due to the fact that I was using museum specimens, but the fact that it occurred in an entire couple of groups was interesting. This is opposed to Myctophidae, where we saw a lot of additional scale lens morphology and very robust and ability to see these different types of reflectors and pigment layers. And then in this specimen here, we have things called double scale lens reflectors and double pigment layers, where you've got two. And there was only one previous study, which was this O'Day study, that had mentioned seeing two different layers. And in mine, I was able to find at least multiple specimens that had double reflector layers and double pigment layers. And so what that means for the group and what that means for the light that's coming out of the organ, again, yet still to be discovered. So here's, and then the robustness of the reflective layers, much easier to see within Myctophidae, much more difficult to see within some of these other subfamilies. And then we're gonna skip that real quick. And I'm gonna talk about one particular fish and we'll kind of move into the conclusions. So this fish, called Scopelopsis multipunctatus, had a much more simplified light organ than all the other lanternfishes. And so these are some images here. You can see it doesn't have that scale cup. All of this photogenic tissue is just embedded into the skin. And it's the only species that doesn't have that scale cup. And so what's going on with Scopelopsis? Scopelopsis is the only lanternfish that actually has a photophore on every single scale pocket on its body. It doesn't really have those large, big photophores. And people that have looked at this fish through ontogeny have seen that when it's a larvae, it starts to create what you would expect to be the regular complement of photophores. But as it gets older and grows larger, it starts to gain additional tiny little photophores along every single scale pocket on its body. And those initial photophores kind of cease to grow. They just don't grow anymore. They cease to grow, they cease to get bigger. And by the time it reaches juvenile and into adult stage, it's really, really hard to discern the differences between the two. But I'm looking at a specimen. This is the first time that this particular species has been dissected and had histology done on its photophores before. I was able to determine one of these scales because I was able to not only dissect it, but find one of those modified scales. And so we know that this is or should be a primary photophore, but yet it still lacks that scale cup. And so Scopelopsis is the only lanternfish that has been found so far where the scale cup is gone on a primary photophore, which is interesting. And so what did we find? Well, we found a lot of variation in density of pigmentation, in the photophore cup and thickness, calcification, and the number of reflective layers and pigment layers. And we found a lot of differences in the length or not the length, but the thickness and the matrix makeup of these modified scale lenses, and also the shape as well. And we also saw that there are multiple instances of these secondary reflectors, including people that have seen it in at least one species beforehand. And that there are patterns in reflector presence, and that there's a unique morphology, at least within Scopolopsis multipunctatus. And so, like I mentioned, some of those next steps is going to be using this data to look at it within a phylogenetic context to see whether or not this has a lot of phylogenetic signal, or whether or not habitat is possibly playing a role in the use of these different types of photophores, but also obtaining fresh specimens, because with fresh specimens, you're going to be able to actually look at different aspects of these light organs, similarly using histology, because oftentimes when you're using fixed and preserved specimens, you lose the ability to see certain cell types through that preservation method. And one of the really interesting things would be to actually look at some of these reflector layers. So the reflector layers are made of something called guanine, and guanine within these layers is actually made up of these tiled little cells, tiled crystals, you could call them. And depending on where the guanine is located, it actually is different in shape. And within fish, when you think of a silvery fish, that's guanine. And if you have multiple layers of guanine, it actually affects the way that light attenuates and bounces off of that fish. And so by gaining fresh specimens, we can actually look to see if the lanternfishes that have two reflector layers versus just one reflector layer maybe have differences in the morphology of their guanine as well. So with that, I'd like to thank all of the institutions that helped and donated specimens and the funding that I received, and I'm happy to take any questions.
Questions for Rene?
Yeah. Surprisingly easy to get fresh specimens of some species. Oh, yeah, sorry. So the question was, what are the challenges and how easy is it to get fresh specimens? And so that's why this study, although important, also lacks some areas, because fresh is better when it comes to histology. Some species are really easy to get, right? They are super abundant. They come to the surface every night, but there are other species that are much more rare. They live deeper. They don't always come to the surface. Like the fact that they all migrate is actually the caveat, right? Like most of them migrate. And so pretty easy to get fresh specimens of a bunch of different species, but you've got at least a third of lanternfish that it's extremely difficult to get species. And so this study, by using museum specimens across all of the genera, I was at least able to compare the morphology of all of these are fixed and preserved, even the ones that are rare, as opposed to the ones that are fresh. But now that we're seeing some differences, getting those fresh specimens, especially in much more easy to get ones, should be pretty easy. But it will be very difficult with the much more rare species. So the question was whether or not we see bioluminescence evolving in inland waters and in deep lakes. And it is not ever, there's no other species other than marine species that bioluminescence has been seen in at least water systems, right? Hypotheses as to why, it is partially due to the fact that if you have inland fish that, let's say, live in that deep sea lake system where they have started to evolve, like they've moved down, they've evolved, they've either lost pigmentation, lost eyes or whatever, we still think that it might still take more evolutionary time to potentially evolve the ability to bioluminesce, or it really truly does rely on a system that has food sources that have some of these luciferin and luciferase compounds already there. We're not sure, but that is one of the possible hypotheses as to why you don't see it in freshwater deep systems.
So just to clarify, the photophore is a lens from which light emitting
So he's just clarifying what a photophore is. So no, a photophore is the light organ itself. It's the whole thing.
Okay.
The lens is just a part of a photophore.
Okay.
Yeah. So the photophore is much more complex light organ. Light organs have a wide variety of anatomies and morphologies. The photophore, at least within lanternfishes, is that specific lens occluding light organ. That's the whole thing. Yeah.
That some species actually use this for communication, but it's just hard to see that and have that. Is there any way to make it part of the ecosystem of this, or is it too hard to get their migratory pattern?
So the question was, are there ways that we could potentially create an artificial habitat in order to see communication via bioluminescence? So we have seen, I should have made the mention that we do see communication via bioluminescence in those ostracods, for example, where they have specific courtship patterns that are flashing. It's just those are more nearshore systems. And so it's very difficult to see in the deep sea. And so to answer whether or not we can create a modified habitat, very hard. Again, because like you mentioned, these fish migrate up and down in the water column. They do not exist in a world where they're used to like not hitting things. And so by containing them, you can actually keep a lanternfish alive for a little bit of time, but it's not used to that system. They tend to bang into aquarium glass very often. And so it's very, and not only that, but then within this closed system, how can you know whether or not the flashing they're doing is to each other? Is it due to stress? Is it due to any one of confounding variables that may occur? And so very hard. Yeah.
Or is you'd have to be like a submersible and it's very expensive, and would that actually improve your research? Go down there if you have a really good grant to do that.
Right. So the question was, have I been to the deep sea? Can I go in a submersible? How do I do my science? So yes, people have gone down into deep sea submersibles. It's very expensive. So that is one major, I guess, barrier to doing research, looking at behavior specifically in the deep sea. So people have put cameras down. They'll have camera traps, but usually they have light sources that then look at things that come into a food trap and things like that. When you shut off all the lights, you can see flashing, but you still can't necessarily see what is flashing, how they're doing the flashing. And so if I had a grant to like go down in the deep sea and look at this behavior and I had good enough cameras that could not only capture the flash, but capture the fish in the dark as well, oh, I 100% would do that. It's like my goal. I don't know if you know Nick Kramer, the fisheries podcast, but he asked the question at the end, what would you do if you had them in limited amount of money? And that is the exact answer I gave him. I would love to go to the deep sea using a submersible, look at these things. And so that's why when I kind of talked about this a little bit, you have to have the caveat of all of this is hypothetical, right? Like, yes, I can show you how the cellular makeup of this organ is, but how it's used behaviorally can only be talked about in the context of, oh, okay, well, these species have sexual dimorphism, and we really only see sexually dimorphic organs or sexually dimorphic things if you have differences between males and females and that they can kind of recognize that. So we infer from other systems to the deep sea oftentimes with this types of stuff. So it's hard. But you do a lot of morphology and you do a lot of inferring and then you just reiterate or revise as you get more data. So, and I've been deep sea trawling. So I've some of the pictures that are on there from the deep sea trawl that I've gone on, but not in the deep sea. I would love to go in the deep sea. So the question was, where have I been getting specimens from? So in this study, and I didn't mention this, but oftentimes, so the natural history museums that you see across the world actually have been collecting lanternfishes for hundreds of years, like literally hundreds of years. And since they're one of the most abundant vertebrates on the planet, they usually have a lot of them. And so oftentimes I'll have specimens sent to me, but again, those are already preserved specimens. And some of them are from multiple different oceans across the world as well. And that's where you start to get some of those more rare species where maybe it only occurs in the Arctic or maybe it only occurs over near New Zealand. Oftentimes you do have species that are very global in their distribution, but you do have species that are much smaller and have they're less abundant. And so that is a good way to get specimens, is to get them from other institutions that will give them to you on loan. And you're like, hey, I need to dissect it. And they're usually like, yeah, that'll be fine. Or, oh no, we only have one of them. We don't want you dissecting it. So you kind of have to play that game as well. Yeah.
Early on, you had a graph that showed time, and you showed a lot of radiation. I can't remember what, like in the Miocene after 20
Oh, yeah.
So was that when a lot of this deviation perhaps occurred? Is that old for the lanternfish? Or I can't remember what that graph is about.
So we're just talking about lanternfish diversification and speciation. So lanternfishes are hypothesized to be about 100 million years old, and people have actually tried to look at their abundance and their diversification associated with not only bioluminescence, but actual oceanic events, right? So the shifting of Pangea and the creation of the Atlantic, is that something that created like a whole new habitat for a bunch of these fish to move into? And so that graph was to show that, yeah, right around 100 million-ish years, you see lanternfishes evolving and diversifying faster than you would expect given that age of 100 million years. And that's a lot of fossil calibration talk that isn't necessarily, that's kind of a whole other topic, but yeah, right around 100. And just the programs that is associated with, I'm happy to talk to you more about if you want. Oh, like up and down or side to side? Oh, so the question was, they migrate vertically in the water column, but how much distance might they migrate like horizontally? I actually don't know that. I know general distributions of specific species. I don't know so much about distance as far as a particular individual in an area. So when they are larvae, they don't really swim. And so you have ocean currents that are moving them mostly, but once they reach an area, from what I know, is they kind of exist in that area. But as far as true distance, not actually sure. Yeah.
Using regular cameras and deep sea camera traps. Is there any possibility of using infrared or different colorway cameras?
So the question was about using infrared cameras in their habitat to record like behavior and things like that. Probably. I'm not 100% sure. It's just one of those. Not only do you have to, so the deep sea, although these fish are extremely abundant, is a moderately depauperate area. And so you have to also get that, you need to get that submersible down, and you need to get it into the deep scattering layer itself, which is constantly moving, right? And so getting it into that area without disrupting what's going on in that area and being able to capture them as they're moving by, that's kind of a huge issue currently, is they do move, and they're not just sitting there, and they do shoal. So I think I can't remember if it was Blue Planet 2, maybe, like you do have individuals of shoaling lanternfishes at the surface, like they do migrate up there, but you can only tell so much. So I mean, possibly that's something we could all look into. Yeah. So the question was about how you are able to control bioluminescence, and also specifically within like defense and predation and things like that. So the ones that have intrinsic bioluminescence, that have the light organs and the cells that can do it, it's innervated by nerves. So they can control it. Yep. They can control when it flashes, how frequently it flashes, all of that. The ones that are the species, the different fish species that have bioluminescence in a symbiotic relationship with bacteria, so like an anglerfish, and there's a caveat to this too, but within that pouch, there is something that the bacteria are doing called quorum sensing. where if you have a certain amount in there, a certain density of bacteria, and they're sending chemical signals to each other, if it reaches a capacity, they'll start glowing. And not only that, but if you, what's the word? If you like shake it, right? Kind of like a snow globe, that agitates them and they start glowing. Kind of like if you splashed around in a wave and you start to see the like the bioluminescence in the waves, kind of the same thing. And so in these fish, yes, they can control it. In other deep sea fish that have symbiotic relationship, they can't necessarily control it, but a lot of those fish have evolved a way to shutter it. So the flashlight fish that we're using it to hunt for prey, a lot of the different species can actually rotate their eye, or they're not their eye, but that pouch down, and they have a pigmented layer that completely covers it so they can rotate it down, or they have shutters that come across. And so fish that have bioluminescence in that symbiotic relationship have evolved different ways to turn it off when they want to, because it's still glowing inside of them that they just block that light. Does that answer your question?
I have a quick question.
Yeah.
So I'm thinking about how you would model this mathematically and thinking about fitness. So is there any trade-off between like fin size and like bioluminescence, or I guess are there any trade-offs?
So
maybe that's
no, it's a great question. So are there any trade-offs in like bioluminescence and other morphological, body, whatever, anatomy-based things? And not a ton has been looked into that, but we, as the people that kind of study bioluminescence, have a hypothesis that you do have a trade-off between body size. So it's not so much the morphology of your body as it is the size of your body. Because if you think about how big all of these things are, most of the fish that are bioluminescent are only like this big, right? Tiny little sardines, maybe up to a foot long, maybe. And so as you get bigger, you have to have more of that chemical compound in your system, and you have to have a big enough light organ and able to either obstruct that light organ or innervate that light organ. And so we think that there is mainly a constraint on the size you can be in order to bioluminesce, because we don't see anything big